Thank you all for joining. I see some familiar names on the call, and I appreciate you taking the time today. This is a collaborative project that I'm going to talk about, and one of the goals is to create a resource for the field. So I would very much welcome any feedback or suggestions, either during the talk or at the end. So the overarching question that motivates our work is to understand how it is that obesity is closely associated with this constellation of serious chronic conditions. And collectively, the conditions listed here, including cardiovascular disease, metabolic disease, and cancer, make up the leading causes of morbidity and mortality in this country. So if we think about how it is that this occurs, it's important to realize a couple of things by way of background. So we know that although obesity is a major health problem, more so than obesity per se, where excess fat is stored is an important determinant of health. And this slide just summarizes many, many decades of epidemiology, which collectively have shown that if you take people matched for weight or BMI, individuals who store their excess white fat in a visceral or apple-shaped location are much more likely to have diabetes, hypertension, cardiovascular disease, cancer, and even mortality. In addition to white fat, we now know that adult humans also have brown fat, and brown fat is associated actually with protection from some of these adverse conditions. And in this slide from work done in my lab, we used a large retrospective study to show that individuals with brown fat matched to individuals without brown fat for a number of key covariates have significantly lower odds of type 2 diabetes, as well as a number of prevalent cardiovascular conditions. And this is true across different BMI strata. And importantly, these associations are done controlling for BMI. So this is body weight independent. And so if we think about how this occurs, that's led my group to study the biology of fat or adipose tissue. And mammals, mice, humans contain two main kinds of fat. There's white fat, which is specialized for storing excess calories in the form of lipid, and brown fat, which can dissipate energy in the form of heat. And these bioenergetic properties of white and brown fat are probably what most people think of when they think of adipose tissue, but it's become increasingly clear over the last years that beyond its bioenergetic role, fat is also an important endocrine organ. And I show you here a number of adipose-derived factors that have been very well studied and are collectively referred to as adipokines, with very famous ones being things like leptin, adiponectin, and adipsin, which are thought to come mainly from white fat. There are also some brown fat-derived factors that are shown here. And these are soluble molecules, either proteins or metabolites, that can act either locally or at a distance to coordinate whole body metabolic processes. And so when we began to approach this question, we were intrigued by the fact that although there are a number of very well-studied fat-derived molecules, such as the ones I showed you on the previous slide, fat cells are actually predicted to secrete more than 1,000 unique polypeptides and probably an even greater number of small molecule metabolites or small peptides. And the vast majority of these, along with their targets and functions, have actually not been characterized. And one of the major reasons for that has been a limitation in technology. And we think that if we can address this problem and build a list of all of the fat-derived factors and begin to deorphanize them or characterize their role, that could allow us to explain really important questions like the links between obesity and disease, perhaps some of the protective effects of brown fat, and ultimately identify new therapeutic targets. And so it was that motivated our establishment of this collaborative group that I'll tell you more about in a minute, with the goal being to establish an encyclopedia of the adipose secretome. And that spans across a number of conditions, as shown here, ranging from, at the most basic level, the secretome of adipocytes in a dish, through to adipose tissue in both the healthy state and in response to physiological stress and in pathological states like obesity and diabetes. We also wanted to characterize the secretome in humans. And then finally, we wanted to move beyond just assembling a list of factors to actually characterizing some new biology of previously unstudied secreted factors. And so to address these goals, we formed a collaborative team, and we've been really fortunate to be funded by an RC2 grant from the NIDDK. And so I'm showing you the other lead investigators on this grant. Each of us has very different expertise. So my focus has been more on adipose tissue biology as well as translational studies in humans. Brian Chait, who's a colleague at Rockefeller, has longstanding expertise in mass spectrometry. David Fenyo from NYU, who is a computational biologist. And then Alan Sagatilian and Alice Ting are both chemical biologists, with Alan's focus being on small molecules, metabolites, and peptides, and Alice's, at least as it relates to this grant, on some of the technology development that we're using in our studies. In my lab, since I'm representing our work, I want to also, just before I tell you about the work, show you the cast of characters who did the work. And this is a large group of really talented people spanning many years in the lab. The first 4 individuals have all now moved on from my lab to bigger and better things. But a lot of the work I'll talk about today has been led by Kaya Plusinska, who's a postdoc in my lab, and also involving Ruiji Jiang, a research assistant in my lab. So let me tell you a little bit about some of the methods we've been using before I get into some of our findings. So I'm showing you here the different approaches that we're taking in this wide-scale project, with the first 2 being the ones I'm going to focus on today: bioorthogonal non-canonical amino acid tagging, or BONCAT, and proximity labeling, or TurboID. And these are both techniques that we use to identify and quantify proteins. We are also doing work focused on metabolites and small peptides, as well as method optimization for mass spectrometry, but I won't have time to tell you about that today. So as we think about proteins, the biggest challenge that we faced is depicted on this slide here, and that is the fact that in the blood, which is what we're most interested in, proteins exist over 10 orders of magnitude in concentration. And so because of that, it's known by many people, sometimes from learning the hard way, that if you simply take plasma and do mass spectrometry, you will get a lot of reads for things like albumin and globulin and these abundant proteins shown in green, but it's very difficult, if not impossible, to identify proteins in the nanogram per mil range or lower, which is where most bona fide hormones exist. And so we realized that we needed to use new approaches to circumvent those problems. And one of the first approaches we used is this technique, abbreviated BONCAT. So the way BONCAT works is shown here. If you think about its simplest iteration, which can be done in vitro, normally when you have newly synthesized proteins, methionine, for example, is incorporated into the nascent proteome by methionine tRNA synthase. And there exist methionine analogs, and we used one called azidohomoalanine, which contain an azide group, which do not exist on amino acids in nature. Azidohomoalanine can be recognized by methionine tRNA synthetase and incorporated into the nascent proteome, which then gives you azide-labeled proteins. And that's very, very important because that allowed us to do some of the experiments shown here. And this is work that was done by Chan-Hee Choi, a former MD PhD student in my lab. So when Chan-Hee started out, he wanted to begin with what we thought was the simplest model system, which was profiling the secretome of the different kinds of adipocytes in a dish. And so he made primary cultures of either interscapular brown fat, subcutaneous white fat, or perigonadal white fat, differentiated them in a dish, And then once they were fully differentiated, he removed the media and replaced it with the media lacking methionine and containing azidohomoalanine. So from that point on, all newly synthesized proteins will be azide labeled. Secreted azide labeled proteins are then pumped out into the conditioned media. An important factor that really is a benefit of this method is because we're only labeling newly synthesized proteins, we can otherwise keep cells in their media, including fetal bovine serum, because those are pre-existing proteins that will not be labeled. And so the reason why this approach is so valuable is because one can then take conditioned media, and these azide groups will form a covalent bond with alkyne moieties in the presence of copper. This is referred to as a click chemistry reaction. And if we take an alkyne group coupled to a bead, we can use that to bind and enrich these azide labeled proteins, pull them out of solution, digest them, and do mass spectrometry. And so Chen-He did exactly that. He identified over 600 unique proteins, and I'm showing you here the nearly 350 proteins that showed enrichment in the secretome of different kinds of adipocytes. You can see in the heat map here, for example, cluster 4 is a group of proteins preferentially secreted by brown fat. Cluster 1 is a group of proteins preferentially secreted by perigonadal white fat. And if we look at pathway analyses, we see to a large extent what we might have expected. So brown fat secreted proteins are enriched in proteins playing a role in axon guidance. And we know that brown fat is very, very heavily innervated by the sympathetic nervous system. Proteins secreted by perigonadal fat are enriched in proteins involved in activating the immune response and complement system, consistent with the inflammatory phenotype associated with this fat depot. We also did some experiments in vivo. And so in these early experiments, what we did is we simply took mice and we injected them with azidohomoalanine. As you can see from the body weight, it seems to be non-toxic. And we then took serum from these animals and did the same click chemistry enrichment. We were able to identify a few hundred proteins, and I'm showing you here the proteins plotted by abundance. You can see albumin at the top, but in red are proteins that are known to be derived from adipose, as well as a new protein of interest, LRG1, that I'll tell you more about in just a moment. In terms of this in vivo experiment, I'm going to show you how we're now approaching in vivo experiments a little bit later in the talk. But with this approach, keep in mind that the azidohomoalanine is not specifically targeted to the adipose tissue. It can accumulate anywhere in the body. In addition, we did these initial experiments in mice on a normal diet, which contains methionine. So the azidohomoalanine has to compete with methionine for incorporation into the proteome. Even with those caveats, though, we were still able to identify a few hundred labeled proteins in the blood. So just to summarize these early studies, in vitro, this technique allows us to profile the secretome in the presence of fetal bovine serum. We detected more than 600 unique proteins, including many that were not predicted to be secreted. In fact, of the total number of proteins we identified, about 2/3 of them have a signal peptide and are thought to be secreted through the classical secretory pathway, but a third did not. And we're really interested in understanding more the logic of how some of these other proteins get secreted by adipocytes. Of the proteins that are differentially secreted across adipocytes, those number nearly 350, and they demonstrate functional enrichment as I described. And in the animal, at least, we can detect serum proteins in the microgram per mil range, including some adipokines. And then finally, in work I'm not going to show you today, with some optimization to our protein fractionation and mass spectrometry pipeline with Brian Scheit's lab, we now can actually detect more than 1,000 proteins in adipocyte conditioned media, including some in the nanogram per mil range like leptin. And so that really highlights, with all of the techniques I'm going to show you today, there's two aspects. There's the technology, which sounds very elegant, but there's really a big role for technical optimization if one wants to get successively deeper into the proteome. So I want to turn now to a little bit of biology, which is one of our major goals. And so if we go back to the data set I showed you from the in vitro secretome, if we're going to actually prioritize proteins for study, clearly we can't study tens or even hundreds of proteins, at least not rapidly in an academic lab. So we had to be thoughtful about how we were going to prioritize proteins for further study. And so the approach we took was to do some orthogonal comparison of different data sets. And so we looked for proteins that were enriched in adipocyte conditioned media, that were at the RNA level highly enriched in adipose compared to other tissues, and finally proteins that we could also detect in the blood of AHA injected mice. And so when we did that, we got a much shorter list of proteins, and we became particularly interested in this protein LRG1, which has been studied before, but had not at the time we began working on it been characterized at least as an adipose-derived protein, although a number of groups have since put out work on its role as an adipose-derived factor. So let me show you now some of our data on LRG1. At the RNA level, as you can see here, LRG1 is expressed by a variety of adipose tissues. The other main place it's expressed is in the liver. When we fractionated adipose tissue from mice, we saw that its expression was very much enriched in mature adipocytes as opposed to the stromal vascular fraction. We were able to show in vitro that LRG1 at the expression level is very much differentiation dependent, and by Western blotting, it is secreted into the conditioned media. So almost all of the LRG1 is in the conditioned media here at day 7. When we thought about its role in vivo, we decided to look at how it changed in the context of things like obesity. And we saw that when we put mice on a high-fat diet, LRG1 levels in the blood went up at multiple time points. And interestingly, at least at the RNA level, this seems mainly to be driven by an increase in LRG1 expression in the adipose tissue. So from these findings, we felt that LRG1 is clearly obesity-related and regulated, and it is an adipokine in the sense that it at least can come from fat, although it does also it can also be produced by the liver. And so to study the physiological role of LRG1, we started with a gain-of-function system using AAVs to drive overexpression of LRG1. We used the AAV8 system, which has relative tropism for adipose tissue. And as you can see here, that led to elevated LRG1 levels in the blood. And at the protein level, most of the increase in LRG1 is coming from perigonadal and subcutaneous white adipose tissues. When we then followed these mice, we were quite interested to see that as these mice were kept on a high-fat diet, the mice that received LRG1 had consistently lower fasting blood glucose, and they had significantly improved glucose and insulin tolerance. And this is despite no difference in body weight. We then did some experiments in a more exaggerated model. So we looked at the DB-DB model, and we used the same adeno-associated viral system to overexpress LRG1. And when we did that, we saw increased levels in the blood as well as the tissues. And interestingly, in this model, LRG1 overexpression actually led to increased weight gain, which we did not see in the wild type setting. And when we looked at these mice, we were intrigued that despite the increased weight gain, they actually had lower fasting blood glucose and lower plasma insulin, at least at early time points, although these differences went away over time. And then most significantly, when we looked, these mice had significantly improved insulin tolerance tests, although a lot of this is probably driven by the lower baseline blood glucose level. So to try and understand what was going on here, we looked a little bit more deeply. We did see that giving DB mice LRG1, as I mentioned, it led to increased weight gain, and that weight gain was the result of increased fat mass, as you can see in these pictures here. And I'm going to come back to this in a minute, but I just want to show you some findings from a reciprocal model where we used CRISPR to knock out LRG1. You can see that the knockout led to a complete absence of LRG1 in the plasma. We also generated heterozygotes that seem to have an intermediate level of LRG1. And when we looked at knockout animals, again, we see no difference in body weight on a high-fat diet. However, now the knockout animals have a reciprocal phenotype with consistently higher fasting blood glucose values. So how is this working? We reason that LRG1 can be produced by adipose tissue, and in obesity, there seems to be an increase in LRG1 production. And how is it that that might promote insulin sensitization? So our clue to how this was going on came from simply inspecting the tissues histologically. And what we saw was this rather striking finding that on either a wild-type black 6 mice on a high-fat diet or DB mice, animals that received LRG1 had far, far fewer crown-like structures. So crown-like structures represent a histological finding that describes macrophages that are thought to be surrounding dead and dying adipocytes, and it's one of the hallmarks of metabolically unhealthy obesity, at least at a histological level. So how is it that LRG1 might be suppressing the accumulation of these crown-like structures in obesity, which have, I should mention, been associated with insulin resistance? So we went a step further and we did some RNA sequencing of these adipose tissues. And what you can see with the pathway analysis and the heat map here is that the largest functional change among genes that were suppressed with LRG1 overexpression were genes involved in inflammation, as shown here in blue. And when we used bioinformatic tools to deconvolute the gene expression, we saw that this is using a tool called CyberSort, which can predict immune cell enrichment based on gene expression. We saw that LRG1 was predicted to result in a substantial reduction in pro-inflammatory macrophage accumulation. And interestingly, that's not just at the tissue level, but it appears to be systemic when we measured blood levels of common inflammatory cytokines like MCP1, CXCL10, TNF-alpha, and interferon gamma. These were all significantly reduced in animals that received the LRG1. So how is this working? How is it that simply overexpressing LRG1 can suppress obesity-related inflammation? And it turns out that when we went back and looked at old literature, LRG1 has its name. It stands for leucine-rich repeat. And it has its name because it has this leucine-rich repeat domain that is specialized for protein-protein interactions. And one of the proteins that LRG1 has been described to interact with is cytochrome C. Cytochrome C, of course, is a mitochondrial protein, and it's known to be involved in apoptosis. So that was initially a little bit of a puzzle. Why should this secreted molecule that circulates in the blood, what possible role could an interaction with cytochrome C have? But what we found, in fact, is that in the blood of these animals, we could show through co-IP experiments that LRG1 does indeed interact with cytochrome C. And so we then developed this model that perhaps in the setting of obesity, when you have dead or dying adipocytes, as these cells die, the mitochondria spill their contents. That includes cytochrome C, which is an abundant mitochondrial protein. And cytochrome C might act as a damp or damage-associated molecular pattern that can activate pro-inflammatory macrophages through toll-like receptor 4 signaling. And so to probe that a little bit further, we looked and we saw indeed that in the setting of obesity and DB or high-fat diet-fed animals, the levels of cytochrome C in the blood go up. Additionally, if we look in vitro at cultured adipocytes, these different modalities of stress, either staurosporin, hypoxia, or TNF-alpha all promoted increased cytochrome C levels in the media. So based on this, we then refined our model further to propose that perhaps in the setting of obesity, the way LRG1 mediates its protective effects is to bind cytochrome C, thereby sequestering it from stimulating pro-inflammatory macrophages. And so we went on to test this. First of all, we isolated peritoneal macrophages, and we were able to show that if we treated those macrophages with recombinant cytochrome C, we could get a very robust upregulation of pro-inflammatory cytokines, and that was blocked when we co-treated these macrophages with TAC242, which is a TLR4 antagonist. We then did the same experiment, adding recombinant LRG1, and you can see that while cytochrome C leads to upregulation of these pro-inflammatory cytokines, at least for 3 of these 4 molecules, that upregulation is very much attenuated when one adds recombinant LRG1. So that we felt kind of confirmed our model that one important way that LRG1 works is by binding cytochrome C, thereby preventing it from binding TLR4 on adipose tissue macrophages and reducing this cycle of tissue inflammation and death. And interestingly, you may have noticed in my prior slides, particularly in the DB mice, that LRG1 gain of function seems to really only affect the insulin-resistant phenotype in a particular time window. And we also see that in our wild-type animals. If you look here, the peak effect appears to be around weeks 10 through 12. And when we looked at this prior study from Strissel et al., we saw that at least on a high-fat diet, this period of maximal efficacy corresponds to exactly when adipocyte turnover is highest. So we believe that when adipocyte turnover is highest in the context of obesity, and there are the most dying adipocytes and therefore the most cytochrome C, that's when having excess LRG1 would be predicted to be most beneficial. So just to summarize here, I've shown you data that LRG1, this protein that we identified through our secretome studies, can bind cytochrome C and dampen its pro-inflammatory effects on macrophages. We believe, based on these findings, that extracellular cytochrome C could serve as a biomarker and perhaps even a trigger for obesity-related inflammation, and that LRG1 in the circulation might act as a buffer against the deleterious effects of cytochrome C release. And so just to show you that schematically, in the setting of obesity and all of its sequelae, we think that LRG1 could be one mediator that can tamp down on obesity-associated inflammation by sequestering cytochrome C. And we're wondering whether it's possible in humans where we have great variation across individuals, might higher levels of LRG1 in the context of obesity be a biomarker for more metabolically healthy obesity. So let me turn now to some ongoing work as well as some future directions. So the focus on LRG1 all came from in vitro studies. And while those studies have been incredibly informative and we continue to pursue them, what we really want to do, of course, is profile the secretome in vivo. And I'm showing you some obvious reasons for that. First, it's difficult to model pathophysiologic conditions in vitro, things like obesity, fasting, or cold exposure. Secondly, as useful as these in vitro systems have been, there is some cellular heterogeneity and there is some artificiality of these in vitro differentiated primary adipocytes. And then finally, and probably most importantly, just because we can identify a factor in the conditioned media of adipocytes in a dish does not necessarily mean that that factor circulates in the bloodstream, which you would need to see if you were going to argue that it works in an endocrine manner. And so to achieve that goal, we've been using both BondCat and TurboID, and this is really the work of Kaya in my lab. And I'll just take you through some of the emerging work we're doing in this area. So the BondCat approach can actually also be used in vivo. And the way that works is shown here. So there is another methionine analog called azidonorleucine. You'll notice, if you remember the chemical structure of AHA or azidohomoalanine, that this molecule ANL has a bulkier side chain, and that normally prevents it from being incorporated into the proteome by methionine tRNA synthase. But what was shown in this work that came from Aaron Schumann's lab is that if you start making point mutations, if you make this single amino acid mutation L274 to G in the methionine tRNA synthase, it can now incorporate azidonorleucine into the nascent proteome. So based on those findings, they generated a conditional mouse model that expresses this mutant methionine tRNA synthase in a Cre-dependent manner. And they were kind enough to share those mice with us. I think they're now available at JAX for those who are interested. And so we've crossed those mice to different Cre lines, to either adiponectin Cre to have the ability to label proteins in all adipocytes, or UCP1 Cre to label proteins in thermogenic adipocytes. And so what one can now do with these conditional mouse models is that protein labeling will only begin once ANL is administered to the animal. And from that point on, newly synthesized proteins only from those cell types will be azide labeled. And then one can couple that with a physiological perturbation like chow or high-fat diet, cold exposure, or exercise, to name a few, and can isolate blood from these animals and use the click chemistry reaction to enrich those proteins derived from adipocytes and identify them by mass spectrometry. In principle, these techniques can also be used not only to identify adipose-derived proteins, but also to trace them to their tissue destination to identify their potential targets. So just to show you that this works, here's an example of a UCP1 Cre crossed to this mutant methionine tRNA synthase strain. The way we do this to optimize labeling in vivo is we do put the mice on a low methionine diet to deplete methionine so there's no competition. And if we couple ANL administration with either housing at thermoneutrality or in the cold, you can see that only Cre-positive mice have really nice labeling of nascent proteins in the brown fat with a very low background signal in Cre-negative animals. So what we can now do is take blood from these animals and identify cold-induced secreted proteins in the bloodstream of these animals. So in addition to BondCat, we're also using a variety of other approaches. One is called TurboID, and this relies on proximity labeling, which is a technique that was discovered and described by Alice Ting's lab. So this involves the engineering of a bacterial biotin ligase to create this supercharged form, if you will, that they call TurboID. And so this enzyme adds a biotin to proteins in close proximity. And the enzyme can be targeted to the endoplasmic reticulum, where it would specifically label proteins in the ER secretory pathway. And using a conditional mouse model that only expresses TurboID KDEL in a Cre-dependent manner, we can now again label proteins in a cell type selective manner and couple that to a physiological stimulus upon giving the mice biotin. And so we have those mouse models, which we're using for a whole variety of studies. Although I've shown you all of the virtues of doing cell type selective labeling, we are also doing some studies with bulk serum or plasma. While we can use those for proteomic work, we're also using those tools for things like lipidomics and metabolomics. And then we also are really interested in not just working with cellular and animal models, but also working with humans. And so we've done 2 studies that I'll tell you a little bit more about now, really focusing on the brown fat side of things by doing cold exposure followed by deep plasma profiling. So in the first of those studies that we call the cold vest study, We recruited 20 young, healthy subjects at an age and BMI where they were very likely to have cold-induced brown fat. We tried to exclude people with any underlying conditions or any medications or illicit substances that might confound the interpretation. And so what we did was we used a customized cooling protocol following methods that were developed by Aaron Sipis and colleagues at NIDDK, where we did some phenotyping and collected blood at baseline. And then we put these cold vests on the subjects and we successively lowered the temperature of these vests until they started shivering, and we defined that as their shivering threshold. We then raised the temperature by 2 degrees and maintained them at that temperature over 3 hours. At the end of the 3-hour time period, we sampled blood again. And so with those samples, we've done extensive plasma profiling, and this is also a highly collaborative effort, including Robert Gersten at Beth Israel and Alan Sagatilian at the Salk. So in humans, of course, we don't have the genetic tools to do cell type selective labeling. So we've done proteomics using 2 commercial platforms, SomaScan and Olink. We've also done metabolomics and lipidomics to allow us to identify factors regulated by the acute activation of brown fat. And we can then, of course, intersect those data with our mouse and cellular data to identify candidates to prioritize. And so we're now completing this first study, and I just want to show you a bit of data since the work's not published yet. This just is a volcano plot of our plasma metabolomics from these studies. So in blue are metabolites that are upregulated, and in red are downregulated. We were really pleased to see that one of the metabolites that was consistently upregulated in subjects with cold was dihome, which has been described by Yuat Seng's lab as an important brown fat-derived lipokine. We also were pleased to see this metabolite DMGV as being downregulated. This has been studied by our colleague Robert Gersten's lab, and it is positively associated with incident diabetes and fatty liver disease. So the fact that it's downregulated by cold exposure is consistent with some of the metabolic benefits seen with brown fat. We also are looking at chronic cold exposure. And so to do that, we're doing more of a study in the wild, and we call this the MIWI study, or Minneapolis Ice Water Immersion Cohort. And so in this study, we're looking at a few hundred people who voluntarily swim in ice water during the winter in Minnesota. And so what we decided to do is to phenotype people either during the winter when they're doing this cold water immersion, or in the summer when they're not. And then as controls, we have friends and family who live in the same environment but don't practice ice water swimming. This is all in collaboration with Betsy Sequist at the University of Minnesota. And we have done phenotyping on these individuals and are now in the midst of doing the same sort of deep plasma profiling. And we're particularly interested to overlay these data with our study of acute cold exposure to see what is in common and what is different. So in the last few minutes of the presentation, I just want to tell you a little bit about the data resource side of things. So this effort has been led by David Fenyo's group at NYU. And so we have built a website that's very much a work in progress that we call Secretopia. This is the web link secretopedia.org. And I just want to show you some of the functionality that we currently have up and running, and it's continually being improved with data being added as we're doing experiments. So if you go to this website, you'll see a number of tabs. And if, for example, you click on the data tab, that will display different data sets that are available to you to view. And if you click on a given experiment, so here, for example, is an in vitro experiment where we've done BONCAT comparing primary adipocytes from either wild-type mice or PRDM16 knockout mice. This is a knockout of a transcription factor that's required for thermogenic fat. it will give you this volcano plot of all of the proteins in the conditioned media that are either up or down. There is some annotation based on whether they are known or predicted to be secreted by different algorithms. So all of these data sets are searchable both in a volcano plot or in a tab delimited format version. It's also possible to do searches on specific proteins. So I'm just showing you here one example. So there's a search box where you can type in the abbreviation for your favorite protein of interest. And so here's AdipoQ or adiponectin. And if you do that, it will highlight where that protein is on the volcano plot in our wild type versus PRDM16 knockout experiment. It's not different. We didn't expect it to be different. And it will tell you some information about the protein, including whether or not it's predicted to be secreted using these different secretion prediction algorithms. And then that can also be done for a variety of proteins, both well-studied and not. And you can look at this in a tabular fashion if you want to see, according to different well-studied algorithms, whether or not your favorite protein is predicted to be secreted. And then finally, something that's in development, but we hope to have running smoothly soon, is this tool for integration. And so what this tool will allow one to do is to type in the gene name or protein symbol of one or multiple genes or proteins of interest, and then you can choose which experiments you want to look at, or even all of them. And then you can see how that protein behaves across a spectrum of experiments. And in addition to all of the proteomic studies we're doing under this grant, we've also uploaded a number of RNA sequencing data sets from my lab. And so the goal ultimately is to be able to integrate transcriptomic and proteomic data on a large scale. So just to close, some of our ongoing efforts and goals are to finish building this encyclopedia of the adipose secretome in mice in both normal physiology and in response to different states of stress and in pathological conditions, to functionally characterize novel secreted molecules in obesity, diabetes, and metabolic diseases. I showed you work on one such molecule, and we have work underway on a number of others. We're also interested in doing this in humans, not only in response to cold exposure, as I told you, but we're gearing up to start some studies on people with insulin resistance, obesity, and type 2 diabetes. And then we also think that another important aspect of this work is on the technology side, so developing new and optimized methods both to enrich proteins and to identify them by mass spectrometry and ultimately identify their targets. And I hope I've convinced you this is worthwhile and can have wide impact. We think that the technical advances can be useful across many fields. And of course, the individual molecules and biology we uncover could be very relevant for obesity, diabetes, and other metabolic diseases. And this can also be applied to any cell type or tissue. And so one day it may even be possible to think about building an animal-wide interactome. And so more long-term, of course, because we really want to contribute to improving human health, we're hoping that we can identify and characterize some novel adipose-derived factors that can be used to ameliorate different obesity-related complications. So I've tried to acknowledge different collaborators along the way. These are the people in my lab. I highlighted the people in my lab who've led our efforts so far. We're really, really grateful to the NIH and NIDDK for supporting this work through an RC2 grant. I want to also particularly thank Carol Haft at NIDDK, who's been a wonderful source of advice and guidance as we've moved forward with this project. Former lab members and then collaborators who I named throughout the talk. Thank you for your attention. I'm happy to take any questions.
Okay, great. Thanks for Paul for the great presentation. So now is our question and answer time. So you can unmute yourself or just type the question in the chat or raise your hand if there's more people. Yeah. So, okay, there is one question from Swapan. Is SAT and the white adipose derived secretome different in human? How you study that?
Yeah, so great question. The short answer is right now we don't know. So the way we're thinking of, at least to partially address this question, is with human subjects, we can of course analyze the proteome of their blood. And then in one of our protocols that's going to begin soon, we also have included in our IRB protocol performing subcutaneous fat biopsies. So of the proteins we identify in the blood, we can at least look to see which of those at the RNA or protein level are abundant in subcutaneous fat. Visceral fat, of course, is not as accessible, so we don't have a direct way right now for how we're going to address that.
Okay, does that answer your question, Swapan? Yes. Okay, so the next question, I think Yuhua, you have a question, you raise your hand.
Yes, hi, Paul. That's fabulous, very exciting data. I have a question about the LRG1. So I may miss it. So do you think it is induced by high-fat diet, and it's a way that, or it's induced by obesity? So I'm thinking that it is like a signal the fat cells sense obesity. So secrete this factor to counteract the obesity-induced inflammation.
Yes.
And yeah, so you
Yeah.
can.
Sorry,
Yes.
I didn't mean to cut you off. So thanks, Yua. We think that this is a factor that is secreted at higher levels in response to obesity and not necessarily a high-fat diet. And the reason why I say that is because we see elevated levels of LRG1 in the blood, not only in wild-type mice on a high-fat diet, but also in DB mice, which are on a standard chow diet. So we think it's the obesity more so than any diet. And there are some human studies where LRG1 has been shown to be a biomarker in people with obesity. And so initially, based on those studies which reported higher LRG1 levels in obesity, when we started the studies, we predicted that LRG1 would actually be a deleterious factor. But the studies we did that I showed you actually suggest that, if anything, it's protective.
So then in those unhealthy obesity people, right?
Yeah.
Do you think that it's a failure of LRG1 to counteract the informations to bind to the cytochrome C, right? Yeah, I think that's the mechanism.
Yeah, so that would be the prediction. We haven't done those studies in humans, but that would be the prediction. We would presume that just because of the heterogeneity across humans, that there's probably a lot of variation in terms of how much dead or dying adipocytes they have, and in turn, how much cytochrome C they're spilling out. And then there are probably also variations in how much LRG1 they have, and all of those things could be important. The other thing I want to emphasize is that although we focused on LRG1 in its role as binding cytochrome C, that doesn't rule out interactions with other proteins, either other damage-associated molecular patterns or in other systems that's been described for its role in modulating TGF-beta signaling, which could be another mechanism underlying its role.
Thank you.
Thanks.
Okay, so next question is coming from Lynn.
Hey, Dr. Cohen, this is Lin Xia from Jocelyn Diabetes Center. I really enjoy your talk. I know you primarily focus on adipocyte-secreted proteins, small peptides. And we also know that adipocytes is the major source for lipid secretion. So I was wondering if you could comment on adipocyte-secreted lipids, for example, the signaling lipids, in regulating the whole body metabolism.
Yeah. Hi, Ling. Thanks for your question. Yeah, it is something we're looking at, and that work is mainly being done in conjunction with Alan Sagatilian's lab. So I didn't have time to talk about it here, but we have done lipidomic profiling of cells in vitro, as well as tissues and blood from animals. And then we also have done this in our human subjects in the cold exposure studies where I showed you that bit of data towards the end. The major challenge we face with metabolites and lipids, unlike proteins, is that there is no technology we currently have available, nor that we've been able to envisage, that allows us to label metabolites or lipids in a cell type selective manner. So we can certainly identify them in the blood. But then the next natural question is, well, how do we know that it came from fat, or how much of it came from fat? And that's a much harder question to answer. One of the things we're doing, which Yua's lab has done very nicely with some of these brown fat lipokines, is to integrate our lipidomic data with transcriptomic data, for example, so that if we see a lipid up in the blood, for example, with cold exposure, and also see coordinate RNA changes of the biosynthetic enzymes that make it in fat, then we feel at least more confident that it might be coming from the adipose tissue.
Yeah, thank you so much.
Sure.
Okay, so our next question coming from Anne. So Anne's question is the lipolysis regulated the release of exosome with lots of protein. Have you looked at this?
Yes, great question. So we haven't yet done work on exosomes, but it's something people often ask about. There is a lot of work showing that adipocytes and adipose tissue can release exosomes, and that might be an important modality by which fat cells signal. We also, in our in vitro study, since a third of the proteins we identified are non-classically secreted, it raises the question of whether any of them are in exosomes. So our main factor that's stopped us, quite honestly, has just been bandwidth to take this on. Not having worked in the exosome field before, I know that properly isolating extracellular vesicles is really, really important. So I think we would want to make sure we were doing that correctly. But the tools I described, particularly BondCat and TurboID, should certainly be usable for exosomes. So one could, in theory, take these different cellular animal models, label proteins in a cell type selective manner, and now go a step further, where rather than just looking at plasma, you could actually isolate extracellular vesicles and then do your enrichment. So I think these tools could be adapted to that purpose. We just don't have any information as of yet.
Okay. Does that answer your question, Anne? Sorry, I couldn't unmute you. Yes. Okay. So are there any more questions from the audience? Okay, there's one from Brian. Can BondCat be used in tissue explant, or is this only used in isolated cells?
Yeah. Thanks, Brian. That's a good question. We have used it in tissue explants, and it can work. And I guess that brings me back to Swapan's question. So it could be that one way to address the subcutaneous versus visceral secretome would be to work with tissue explants. We've done experiments with mouse explants, and if we add the modified methionine, it can be taken up by the tissue and incorporated. Our main reason for not pursuing this approach thus far has just been our finding, and this is seen in the literature too, that the process of making explants leads to tremendous alterations in gene and protein expression, mainly in pro-inflammatory mediators, presumably because the tissue has now been devascularized and denervated. But I think with those caveats, if one accepts them or controls for them, it could still be a useful way for studying explants.
Okay. Okay. There's one question from Carol. Are you aware of any progress on getting a handle of microbiome secreted metabolites proteins that impact adipose biology?
Yeah, so also a really interesting question. There's interesting work showing that the microbiome can regulate whole body metabolism as well as adipose tissue physiology. But I think the black box there, of course, is how does that work? And is it mediated through soluble factors, or could it be neuronally mediated, or both? That is not something we have done any work on, but I am aware of some groups working in that space. I guess I'm not divulging any secrets because he gave a seminar at Rockefeller just a couple of days ago, but Christoph Theis from Penn is doing some really fascinating work uncovering microbiome-derived mediators that impact on different metabolic processes. And so I think he's well on his way to answering those sorts of questions.